In 2023, a team of geneticists published a study that changed everything. They extracted DNA from the bones of 27 enslaved African Americans who died more than 200 years ago. Then they compared that DNA to more than 9 million living people. The result was nearly 42,000 modern relatives, including direct descendants. This is not speculation, this is the DNA record. Welcome to Ancestral Roots, Untold Origins. Today, an exclusive update. The Catoctin Furnace sits in the mountains of western Maryland. It operated from 1776 to 1903, producing iron for the American Industrial Revolution. The workers were enslaved African Americans, men, women, and children. When they died, they were buried in an unmarked cemetery. No headstones, no names, no records. For more than 200 years, they remained anonymous until 2023. A team of researchers from Harvard, the Smithsonian, and 23andMe came together. The lead geneticist was David Reich, a pioneer in ancient DNA research. The historian was Henry Louis Gates Jr., director of the Hutchins Center for African and African American Research. The anthropologist was Douglas Ousley, the Smithsonian's leading expert on skeletal biology. Their goal was unprecedented: extract ancient DNA from the bones of enslaved individuals, then find their living descendants. So how did they do it? The team selected 27 individuals from the Catoctin Cemetery. They extracted two types of DNA: mitochondrial DNA, which traces the maternal line, and nuclear DNA, which contains the full genetic code. The samples were processed in Reich's laboratory at Harvard. After months of analysis, the results came back. The researchers compared the ancient DNA to a database of more than 9 million to 3andMe customers who had agreed to participate in research. What they found was staggering. They identified 41,799 modern genetic relatives of the 27 enslaved individuals. Of those, 2,975 were very close relatives. This includes possible second cousins, first cousins once removed, and even direct descendants. Let those numbers sink in. 41,799 living people, connected by DNA to 27 people who died in the late 1700s and early 1800s. The study was published in the journal Science in August 2023. It was titled The Genetic Legacy of African Americans from Catoctin Furnace. For the first time, a direct genetic link was established between enslaved individuals and living African Americans. But the researchers did not stop there. They also traced the African origins of the Catoctin individuals. By comparing the ancient DNA to reference populations in West and Central Africa, they identified two primary source regions. The first was the Wolof people of Senegal and Gambia. The second was the Congo people of Central Africa, in what is now Angola, the Republic of Congo, and the Democratic Republic of Congo. These two groups represent the majority of the African ancestry found in the Catoctin individuals. Within the cemetery itself, researchers identified 5 distinct genetic families. Most of these family groups consisted of mothers, children, and siblings buried together. One group included a woman and 3 children. Another included 2 adult males who were likely brothers. The DNA confirmed what the cemetery arrangement suggested. This was not just a collection of individuals, this was a community. Now, let me explain how any of this is possible. The answer lies in a revolution in genetic technology. Dr. Leo Speidel, a geneticist at the Riken Center in Japan, described it this way. He said, "We can now look back many hundreds of millennia into the past by extracting DNA from ancient bones and applying new statistical techniques that reconstruct the genetic trees that relate these people back in time." A key innovation is called haplotype-based analysis. A haplotype is a set of genetic variants inherited together from a single parent. In the Catoctin study, researchers used a technique called chromosomal painting. This technique breaks each person's genome into segments and assigns each segment to an ancestral population. For the 27 enslaved individuals, the results showed between 80 and 95% African ancestry. The remaining ancestry was mostly European. This European ancestry likely came from the slave-owning population of Maryland. The Catoctin study also used a technique called kinship estimation. Kinship estimation compares the number of identical DNA segments between two individuals. The researchers compared all 27 individuals to each other. They identified parent-child relationships, sibling relationships, and more distant connections. One family tree included a grandmother, her daughter, and the daughter's child. All three were buried in different parts of the cemetery. Without DNA, no one would have known they were related. With DNA, the family was reunited after 200 years. The same technology is being used in other ancient DNA studies around the world. In 2025, a team from the University of Huddersfield and the University of Southampton published a study on the settlement of Sahul. Sahul is the ancient supercontinent that included Australia, New Guinea, and Tasmania. Scientists had long debated when the first humans arrived there. The long chronology suggested around 60,000 years ago. The short chronology argued for 45,000 to 50,000 years ago. The 2025 study analyzed nearly 2,500 mitochondrial DNA genomes. The results confirmed the long chronology. Settlement happened around 60,000 years ago. Now let us look beyond Catoctin to other studies of African origins in the Americas. In 2023, a team at the University of Pennsylvania published a genetic study of African American and Afro-Caribbean populations. They analyzed more than 100,000 individuals and identified specific African ancestral groups. The study found that African Americans have significant ancestry from the Wolof, Kongo, Fon, and Bundu peoples. These groups correspond directly to the regions where the transatlantic slave trade was most active. But here is what history books left out. Individual African Americans often have ancestry from multiple African regions. A person whose family has lived in Georgia for 200 years might have Wolof ancestry from Senegal, Kongo ancestry from Angola, and Fon ancestry from Benin. This mixing happened in Africa before the slave trade. It also happened in the Americas during slavery. Enslaved people from different African regions were deliberately mixed on plantations to reduce the risk of rebellion. The result was a new African American genome, distinct from any single African population, but still deeply connected to Africa. In Delaware, at a site called Avery's Rest, archaeologists excavated a 17th-century colonial settlement. Genomic analysis revealed that African-descended individuals buried there had origins in West and West Central Africa. Even more striking, researchers identified a father and his child buried together. In Brazil, researchers are studying quilombos. These are communities formed by enslaved Africans who escaped captivity. A 2023 genetic study of Brazilian quilombos traced their Y chromosome and mitochondrial DNA markers back to specific regions of Africa. The study also showed far more European male ancestors and far more African female ancestors. A direct genetic record of slavery. In Richmond, Virginia, a different kind of DNA investigation is underway. In 1994, workers discovered a well filled with human remains. The bones belonged to 44 adults and 9 children, mostly of African American descent. These individuals were victims of grave robbing. Their bodies were used for medical dissection without consent. Today, researchers are using genetic analysis to reassociate the bones into individual people. Mitochondrial DNA has revealed African haplogroups, including L1b2a and L1c. Y chromosome analysis has revealed African haplogroups, including E1b1av38 and B2bM112. The researchers are not just identifying bones, they are restoring identities. They are giving names back to the nameless. Now let us go even further back, much further. The DNA record extends back hundreds of thousands of years to a time when multiple human species walked the earth. Neanderthals lived in Europe and Asia from about 400,000 to 40,000 years ago. Denisovans lived in Asia from about 300,000 to 50,000 years ago. Modern humans emerged in Africa around 300,000 years ago. When modern humans migrated out of Africa, they encountered Neanderthals and Denisovans, and they interbred. The evidence is in our DNA. In 2010, geneticist Svante Pääbo published the first draft of the Neanderthal genome. Modern humans of non-African descent carry between 1 and 4% Neanderthal DNA. In 2010, another team led by David Reich published the discovery of the Denisovan genome. Modern humans in Melanesia carry up to 5% Denisovan DNA. These interbreeding events left lasting marks. Neanderthal DNA contributes to skin, hair, and immune system function. Some Neanderthal genes helped modern humans survive diseases in Europe and Asia. Denisovan DNA contributed to high-altitude adaptation in Tibetans. The EPAS1 gene, which helps Tibetans survive low oxygen, comes from Denisovans. The DNA record proves that no population is pure. We are all hybrids. We are all products of ancient encounters between different human species. Recent studies have even found evidence of DNA from an unknown archaic human species in some African populations. A ghost population, one that left no fossil record, only genetic traces. The DNA record is still revealing its secrets. Who has the right to access ancient DNA? What obligations do researchers have to living descendants? The Catoctin study created a new model. Researchers developed a process to notify living relatives. They set up a website where 23andMe customers could check as they matched the ancient individuals. But not every study does this. Most ancient DNA studies publish results without contacting anyone. The individuals remain anonymous. Their descendants never know. In 2021, the journal Nature published guidelines for ancient DNA research. The guidelines recommend consulting with descendant communities, but they do not require it. There is also the question of commercialization. 23andMe is a for-profit company. The Catoctin study required participants to opt into research. Only customers who had agreed to participate were included. This was an ethical choice. The East Marshall Street Well Project in Richmond offers another model. The researchers involved descendant communities from the beginning. They asked the community what they wanted to know and how the remains should be treated. This is community-engaged research. It is slower and harder, but it is more ethical. The DNA record belongs to all of us, but it belongs especially to the descendants of the people whose bones are being analyzed. They should have a voice. The Catoctin study proved that it is possible to do this. Other studies should follow. So what comes next? The next decade will see an explosion in DNA ancestry research. New technologies are making ancient DNA extraction faster and cheaper. In 2010, sequencing a single ancient genome cost millions of dollars. In 2024, it costs a few $1,000s. Dr. Leo Spidel and his team at Riken are developing new statistical methods that can reconstruct genetic trees going back hundreds of thousands of years. They can identify common ancestors without needing ancient DNA at all. The result is a family tree that includes everyone, not just the people who left bones behind, but everyone who ever lived. The Catoctin study identified 41,799 modern relatives. That number will grow. As more people take DNA tests, more matches will be found. As more ancient DNA is sequenced, more connections will be made. Soon, it may be possible for any African American to trace their family back to a specific enslaved ancestor. Not a name, not a record, a person with DNA. That is the promise of this technology. That is the power of the DNA record. Have you taken a DNA test and found something unexpected? Share your story in the comments. Subscribe to Ancestral Roots, Untold Origins, hit the bell, and stay tuned for our next exclusive update.